So without further ado, he is now director of the National Institutes of Health, the whole bunch, not just the Neurologic Institute, but the whole kit and caboodle. So Francis, thank you.
The whole kit and caboodle. Thank you, Anne. And thank you for making that blessedly brief. That's a wonderful way for us to have a symposium and talk about history and talk about science and talk about where this might all be going. I am delighted to be here, not just because it's great to get out of Washington right now, but because this is a very special evening indeed, the 50th anniversary of the Hereditary Disease Foundation, an organization that I've had the privilege of being involved in. Not quite 50 years, but I think I'm adding it up something like 32 years. And the work that has been done through the auspices of HDF and all the advances that have happened have been breathtaking. And yet, as you will hear, we still have more work to do. And that will be part of what we're going to talk about in the next hour as well. When Nancy Wexler asked me to do this, I was honored to say yes. And I thought at the time that I said yes that I would be coming here as a private citizen and not as the director of NIH. Because if you've looked at history, you would notice that NIH directors have invariably turned over when the presidency turns over, and I expected to do that as well. And on January 19th, a day before my letter of resignation was to become effective, I got an email that said, "Your letter has been rejected." There was some consternation and speculation about why that might be. Some of my friends suggested that the grammar must have been unacceptable. But since it was only 2 sentences, I didn't think that was it. And so I was, in fact, pleased and privileged to be able to stay on and to, by this past June, to get a clear indication that the current administration would like me to continue in this role of leading this largest of all the world's biomedical research institutions, the National Institutes of Health. And in that role, I have this privilege of looking over the entire landscape of what's happening in medical research and trying to identify areas that could use a push, that could use some additional resources, that maybe could use bringing collaborators together in a way that would make fields go quicker. And it's that topic of collaboration that I particularly want to talk about here as I go through, admittedly, quick and personal view of the history of the Hereditary Disease Foundation, because it has been all about collaboration. And so much of what was encouraged by Nancy in the formation and management of the Hereditary Disease Foundation scientific group is something that we now have seen adopted in many other projects across the world and across many different areas of science. The idea that you can get things done if people stop worrying about competing with each other and decide to work together. And that has been a contribution that will last for the ages that has been made by this organization. And I couldn't help but put as another sort of title here as I went walking that ribbon of highway, which always seemed to me like that must be talking about DNA. But of course, those are not the words of a scientist. Those are the words of Woody Guthrie, which I'll come to in a moment. But anyway, celebration of 50 years of discovery. Hooray! Glad to be part of this. Yeah, how do you like my animation there? That wasn't easy to do. So the Guthries. I grew up on a small farm in the Shenandoah Valley. My father was a folk song collector. And as you can imagine, Woody Guthrie was one of those venerated names that I heard about from the time I heard about much of anything. And I would often fall asleep in my bedroom up on the second floor of that farmhouse, which had no indoor plumbing, listening to recordings of Woody Guthrie singing some of the songs that he wrote. And of course, I was enormously an admirer of what he contributed to our American tradition, as was my father. I learned to play the guitar because, you know, Woody Guthrie played the guitar. And yet, as we all know, Woody suffered from Huntington's disease, died in 1967. His ex-wife, Marjorie, then established the Committee to Combat Huntington's Disease, which is now the HDSA. And she met with an NIH researcher, Miranthopoulos, working on Huntington's disease, and thus began a partnership between HD and NIH that has continued to this day. You can see the first Marjorie Guthrie Memorial Lecture there, delivered by none other than David Baltimore, which is interesting, our Nobel laureate, who remains to this day one of the venerated wise voices about all of molecular biology. And then in 1968, Nancy and Alice's mom, Leonore Wexler, diagnosed with HD. Milton establishes the California branch of the CCHD, but decides to prioritize basic research, and then that becomes the autonomous Hereditary Disease Foundation. There you see an earlier picture of the Wexler family and a more recent one. And of course, the Wexler family and HDF are very much synonymous in terms of the way in which this whole enterprise has come about. And I may get bits and pieces of this history wrong, in which case I hope Nancy will correct me later. Then, of course, we had to get the country interested. There had to be a way in which support for this condition, which was considered pretty rare and wasn't getting much attention, could be mounted. And so the Congressional Commission for the Control of Huntington's Disease was put in place. And this was a very significant development. Those in the back probably can't see the particular names of the commission members, but that's Marjorie Guthrie as the chairperson. And yes, that's Milton Wexler as the vice chairperson. And who is the commission staff and executive director? Why, it's Nancy Wexler. Do you think the Congress had any chance of saying no at this point? No chance at all. And by the way, the Department of Health and Human Services at that point, called HEW, was also very much engaged in this. And so this was a seminal moment where the visibility of Huntington's disease and the need and the responsibility to conduct research became much more apparent. 1977. And then the HDF came along with Milton's wonderful idea about how to get scientists to really talk to each other. It's still the case, I'm sorry to say, that when you bring scientists together to talk about a topic, unless somebody is really thinking about the agenda, it can degenerate into a circumstance where everybody brings their PowerPoints and they give their canned talk and then they sit down and then they do their email. And nothing much happens. You would never want to do that in front of Milton because he would call you out in a minute. So, no canned presentations allowed. The agenda was utterly fluid. You were never quite sure what was going to happen from moment to moment. And people had to avoid jargon because Milton also wanted to be sure he was bringing together people of different disciplines who would not have understood each other's jargon unless it was carefully defined at the get-go. And always the meetings beginning with a HD family who would talk directly to the scientists about their experience. Many of the scientists in the room had studied this disease by reading about it, had done experiments about it. They'd never actually met somebody who was affected or a family who was taking care of that person. And this was life-changing for all of those people involved. I can't tell you how many PhD students who would otherwise not have had that experience came away motivated and transformed by having met those families. And yes, we were all affected. There's an article there about that Nancy wrote about what this was all about. I first went to one of these workshops, I think it was 1985, yes, and I was a professor at the University of Michigan, actually an assistant professor at the University of Michigan, just beginning to figure out what I wanted to do with my own research. And I think I ran into Nancy at an American Society of Human Genetics meeting, and she said, well, you ought to come to this meeting and talk about what you're doing with your own efforts to find the gene, which were quite rudimentary at that point. And it was one of those extremely interesting and different kinds of ways to spend 2.5 days. We met in Santa Monica under the fig tree. There were these amazing, thoughtful scientists that I would not have normally had the chance to rub shoulders with. And yes, we would always have an amazing Saturday night party where people like me, who were a bit starry-eyed, would encounter people like, Julie Andrews or Carol Burnett or Frank Gehry or whoever it was that happened to be passing by. And that was a nice perk, may I say, for all of us who otherwise were working pretty hard at this, and we're happy to have a chance to let our hair down and have a good time for an evening in between the really hard work that went on at these workshops. I want to give a particular shout out to David Houseman because, in the late 70s, how we were going to get an answer to Huntington's disease, nobody knew what the normal function of this gene was going to be. We knew it ran in families. That much we knew. We knew, therefore, it was caused by a misspelling of a gene somewhere in this big morass of the human genome, about which we knew relatively little, other than there were 23 chromosomes. And so somewhere on one of those chromosomes, there must be a gene that has this misspelling. But that was it. So you're imagining a needle in a haystack. This is worse. This is like a needle in the haystack in the dark with gloves on. How are you ever going to get an answer to this? Well, a strategy emerged And David was very much in the lead of thinking about this, about how to use a genetic technique to locate, in the general way, the neighborhood of the HD gene. And this was basically to use genetic markers, RFLPs. No graduate students today know what an RFLP was, but at this point, these were just variable places in the genome where individuals would have a slightly different spelling. Now imagine you have a big family with lots of people who are affected with Huntington's disease, others who are not, and you look to see, is that spelling being transmitted down through the family in concordance with Huntington's disease? That doesn't mean you found the mutation, but it means you found something close by, or it wouldn't travel through the family in this coordinated way. It must be on the same chromosome. It must actually be fairly close on the same chromosome. So that was an idea which had been done wonderfully well in things like Drosophila, fruit flies, but it hadn't really been tried in humans until about this point. And David proposed that it was time to start. So what was the problem? Well, we didn't have any of these RFLPs. So they had to be kind of invented as you went along. And there were people saying, well, you should wait until you have a complete collection of hundreds of them, because then you have a pretty good chance of sampling the entire genome. And David said, well, yeah, we could do that, but why don't we just start as we're going and see, maybe we'll get lucky. So those markers got started, Jim Gazella pulling them out. In all rights, it should have been, the 195th marker that turned out to be correct, because the odds were really against an early success. But you needed a large family to do this. That was going to help you. And that, of course, was the moment at which Nancy's discovery of this trip and this family on the Lake Maracaibo in Venezuela became critical, because you wanted to have lots and lots of DNA samples to be able to assess whether you were close or not. Well, it was the 8th marker that turned out to be the one that gave this result. I guess, Huntington's disease is a terrible burden for families. It must grieve God to have families going through this. So God figured, I'll give you a break, and we'll give you the 8th marker instead of the 196th, and maybe these yokels can get going on finding an answer to this condition. And there it was. This is part of the pedigree from Venezuela. This was the report, November 1983. I remember when Jim Gazella told this story for the first time. It was the month before that, the American Society of Human Genetics meeting, which happened to be in Norfolk, of all places. You can tell that in those days the society was a lot smaller, because they could fit in a hotel in Norfolk, and now they have to go to Orlando in order to have enough convention center space. But Jim got up. It was one of those sort of late-breaking news, and it was 10:30 at night, and most people had gone to the bar or fallen asleep, and he put up this data, and we all fell off our chairs. Because this was the first time for any disease that this strategy had worked, that you could map the position of an important disorder that's inherited in a genetic fashion by using this set of markers. This was the proof of the idea that this could be generalized. This opened the door to successes that rolled in over the course of the next 10, 20 years of genes that got mapped and then got identified. All started because of Huntington's disease. There's a theme here, by the way, that so many of the things that happened in the field of human genetics started with Huntington's disease, and started because of the Huntington's Disease Collaborative Group, and because of Nancy and her ability to pull all these folks together, and with strong leadership from Milton, convince us that we simply had a lot more to go to get by working together than working separately. And so we did. And here is one of our gatherings. This is not in California. This is in Florida, in Isla Mirada on the Keys, in a wonderful place that we got the chance to use from time to time. You might or might not recognize some of those people. Obviously, Nancy here. Yeah, that's me back there, with sort of peering over the top of the head of David Houseman, who's barely visible behind me. I won't go through naming all these people, but they are all wonderful scientists. They have become lifelong friends. Jill Bates, right here, is sitting there in the 3rd row. There may be other people in the room that I haven't said hello to yet who are in this picture. So this was the nature of the group. And all of these folks decided not to worry too much about who got the credit. We were going to go from Gazella's linkage study, saying this is on chromosome 4, to actually finding the gene. Shouldn't be that hard, right? You're already in the neighborhood? Oh, gosh. It wouldn't be hard now. You know, to go from having an idea where the gene was to identifying the cause. Today, a pretty good graduate student with the right DNA samples and a thermal cycler, I'd give them a week. Yeah, okay, we weren't stupid. We just didn't have the technology, and it took us 10 years. One of the things that my group did was to try to amplify the speed with which you could travel from that linked marker, that G8, which is officially called G4S10, towards the Huntington's disease gene, without having to walk painful step by painful step along the chromosome, which was the traditional way to do it, but to jump and get there faster. And maybe we helped a little bit in that regard. We did a few other things as well, but it was an incredible learning experience. And yes, people wrote little news and views pieces about so near, yet so far. 1990, why haven't they found it yet? By this point, other genes were beginning to get not just mapped, but identified. My own lab finding the cystic fibrosis gene, and well, hey, why can't you find Huntington's if you can find CF? That must have been at least as hard. 1992, the quest goes on, and people still confronted with perplexing data in an unusually difficult region of chromosome 4. We will all agree it was an unusually difficult region of chromosome 4. But, bang, April 1993, finally found a picture of Jim and his folks from the lab, especially Marcy McDonald. And it did finally emerge as a gene that turned out to have an immediate, obvious cause of why it would be responsible for a disorder. Namely that it had within it an expansion, sort of an accordion that got bigger and bigger, of a series of DNA letters, CAG, 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 which normally should not happen more than 20 or so times in a row. But if it got to be 30 or 40 or 50 times in a row, the protein that was made from that was apparently toxic, and that was emerging as the cause of this condition. All of that falling together in April of 1993. And yes, the paper that described this, again, showing that this is a trinucleotide repeat, expanded and unstable, with lots of support from lots of places, as you can see here, with much support from HDF. And this, I think, was the first major publication of the discovery of a gene where the author list was that, the Huntington's Disease Collaborative Research Group. You had to go to the footnote to find out who these people were. And that was intentional. That was to document the fact that this really was a collaborative group. This wasn't people jockeying so that when it finally came out, somebody was going to get the credit and everybody else would get pushed aside. So the asterisk, which I'm sure you can't read, is all these folks who were part of that publication. The 6 groups in the US and in England that together had made this possible. And who, believe me, at that point had a great party. And it was time. Now, I just want to say again how much this discovery and all of the steps that it took to get there inspired not just people thinking about Huntington's disease, but people thinking about anything that related to genetics or to the human genome being the entire instruction book for human biology, 3 billion of those DNA letters. Yeah, we had messed around with a great deal of difficulty in a modest part of chromosome 4, and it had been really hard to do that. If we wanted to take that success and begin to extrapolate it to the thousands of disorders that were probably going to be more difficult than Huntington's disease, they weren't going to be inherited in such straightforward ways, we had to have a better database of information about the genome. We needed that kind of reference. And so, in all those discussions, going back to the mid-1980s and moving up until 1990, when the US Human Genome Project got underway, Huntington's disease was regularly referred to as an example of how we were both learning about how to travel around the genome, but also how we needed better ways to do so if we were going to extrapolate beyond a few disorders and really take on all of the conditions that are affected. And certainly, Renato Del Becco made that point about cancer. Others made that point about complex diseases like diabetes. The momentum began to build. And articles and reports got written, and here are 2 of the reports. The National Research Council, Mapping and Sequencing the Human Genome, the OTA report, Mapping Our Genes, both of which recommended starting a genome project. This is the report of a hearing, November 9th, 1989. I remember this one because I was one of the witnesses. Nancy was one of the witnesses. Jim Watson was one of the witnesses. There was a hapless person from the Department of Energy who was one of the witnesses who got asked what they were planning to do about ethics and had no answer, and that didn't go so well. Perhaps at that moment, Jim Watson decided the NIH project was going to do a lot with ethics, and a good thing that was as well. Who was the main inquisitor sitting at the senator's desk asking the questions at this hearing? It was Al Gore, who became incredibly interested in Huntington's disease as a specific example, but in the Genome Project as a general potential solution to finding answers for lots of different disorders. And so the Genome Project got going, and I'll skip over 13 years of what was pretty intense effort to say, hey, it worked, but so much of what we did to get the Genome Project to work involved building upon things we had learned on a small scale with Huntington's disease, and which then became applied on a larger scale. And many of the people involved in the Genome Project were people who had cut their teeth on Huntington's disease. Just some examples. A very important one, give the data away. When we started working on Huntington's disease, the idea that you would actually share your data with these other 5 groups when you just generated it like the week before and you had a really cool idea and you kind of like to have a little time to play with it, nope, not going to happen. You got to be completely open about this. We are really all part of the team. What we decided with the Genome Project, the whole world's part of the team here. This is our shared inheritance for human biology. And so, a decision made in 1996 that as the DNA sequence of the human genome began to appear, base pair by base pair, we were going to give that away every 24 hours, putting it up on the internet, letting anybody who had a good idea start to work with it and figure out what to do and how to make sense out of it. This is, by the way, a picture of the whiteboard where that decision was made. I think that's probably John Sulston's handwriting, but I'm not sure. It could be Bob Waterston. It was one or the other. As we had that discussion in Bermuda, at this point, I had the job of leading the Human Genome Project on the US side, and I was a big proponent of this, but there were some in the room who were really uneasy about doing this, but ultimately it was agreed. By the way, none of us had any particular authority to make that decision. None of us were representing ministers of health or science. Maybe it's just as well in these big projects that you don't go to that level and let scientists work it out, and that's what we did in this case. And the ethical, legal, and social implications, which Watson had put forward as an important part of the Genome Project. Another thing that I think we did learn from the Huntington's Project, that if we were going to go through this process of identifying the actual cause of the disorder, that meant that there were people who were at risk because of having an affected parent who might now be able to find out, long before any symptoms appeared, about whether they had in fact inherited that misspelled gene and would have a 100% chance of developing the condition if they lived long enough. And that was not possible until the gene was in hand, or at least not directly possible, and now it was. And so that kind of debate about the right to know and also the right not to know began to emerge in lots of other circumstances. And so as this LC program, as it was called, for the Genome Project, got formulated as an integral part of the Genome Project, something that had never been done before, anticipating these issues in advance, who else would we want to have as the overseer of the working group wrestling with those issues than Nancy Wexler, who took that on with great energy and vision and turned what had been a sort of diffuse area of uncertainty into a very explicit set of goals and efforts. Funding training, research, workshops, centers of excellence, and over the course of a number of years, pointed particularly to the issue of genetic discrimination. Again, coming back to Huntington's disease, if somebody has, in fact, a risk of developing this condition, is that a reason to take their opportunity for health insurance or for employment away if they are entirely normal at the time? The answer should be no. And yet it took a great deal, 16 years, of dealing with the Congress before finally in 2008, the Genetic Information Non-Discrimination Act was signed into law by President George W. Bush. And it became no longer a source of concern in the United States about whether your employment or your health insurance were going to be taken away if you turned out to be at risk for a genetic disorder. Now, since the Genome Project got its conclusion in 2003, a whole lot of other things have happened in genomics. And I could give a many-hour lecture on that, and I won't, and you're glad about that because we have dinner. And we have some other speakers who are going to tell you a lot of interesting things about where we specifically are now with Huntington's disease. But I could say all of these projects, which were also sort of large-scale, big science efforts to try to empower individual investigators to go faster with their own research, which is what the Genome Project did. They also were built on this same ethic, that if you have the chance to bring investigators together from multiple different perspectives and figure out how to talk to each other and learn each other's disciplines, and then make sure all the data gets given away, that's a good thing to do. And that's very much what we have been focusing on a lot of our time in the NIH since that time with these projects and others. Not listed up here, but the BRAIN Initiative, now in its third year, an effort to bring together neuroscientists, physicians, computational experts, robotics experts, and a whole bunch of engineers, is another example that I think will be profoundly important for every kind of neurological condition going forward, because we aim to understand how the circuits in the brain do what they do and how sometimes things go wrong. Another sort of, I think, standing on the shoulders of the Genome Project, which in many ways stood on the shoulders of the Huntington's Disease Collaborative Group. So today, very quickly, in 1 or 2 slides before I sit down and let the present-day researchers who are working on this talk to you, there's a lot of things going on in Huntington's disease research, and I am at this time more of an observer and a person cheering on the sidelines. My own lab doesn't currently work on HD, but I certainly look at the papers that come out with a great deal of interest. So basic science. Why does that trinucleotide repeat? That CAG, actually CAG codes for glutamine, so that means the protein that it makes has glutamines in a long string. Why does that cause damage? And why is that damage so specific to the part of the brain that is most affected in Huntington's disease, the basal ganglia? Could we understand how Huntington's disease does what it does by using this amazing set of developments in terms of stem cell research so that we could mimic in a cell culture what goes on in a person? The induced pluripotent stem cell approach, which means I could take a skin biopsy from any of you, take those cells from the skin, add to it an appropriate cocktail of 4 carefully chosen genes, and convince your cells to become like pluripotent, like they could do anything. And then encourage them again to go down a pathway to become neurons, and then see how those behave. And how would that be different if that skin biopsy came from somebody with Huntington's disease? That work is going on. It's likely to be quite revealing in terms of giving us fundamental information about what goes on with that trinucleotide repeat. Genetic studies. Besides the length of the repeat itself, which is a strong predictor of age of onset, the longer the repeat, the more toxic the protein is, the earlier the onset. But that's not the whole story. What else modifies the age of onset and could therefore be a potential drug target? Because if you could make the age of onset 130, well, then that would be just fine. You could call that a cure. And in fact, a lot of work done on this with programs like Track HD, and Sarah may say something about this, to try to identify by studying lots and lots of individuals what other modifiers are there. Recent data suggesting a gene called MSH3 might be in that regard, and that might teach us some interesting things about therapeutics. Clinical studies. If we want to test therapy, it would be nice to have not only clinical progression to look at, but biomarkers, that is, things you could measure that would tell you, hey, this is working, without having to wait for years to find out. And a recent discovery that something called neurofilament light chain seems to be a pretty good correlate for the rate of progression, suggests that it might also be a good biomarker for clinical trials. And I just saw some of that data, and it looks pretty interesting. New imaging approaches so that you get an even more refined view of what's happening in the brain as a result of Huntington's disease. And then this search for new therapeutic options, because that's where everybody now wants to see the success come forward. We have a fair amount of information about these pathways. Can we come up with a strategy that works? New drugs, new ways to affect the DNA, which is where the mutation is, or the RNA, which is made from that DNA, to basically deal with this expanded trinucleotide repeat. Those things which are nicely put out in an article that Sarah and Edward Wilde wrote, which I just stole this particular image from. If you want to really read about the state of the art here, therapies targeting DNA and RNA and Huntington's disease, this would be a great place to look. Just very quickly to show you the diagram, I'm sure Sarah will do much better at this. Now, here's the gene on chromosome 4. There is that expanded CAG repeat in the copy of the gene that causes Huntington's disease. That's the bad actor. That's what you want to try to deal with. Maybe you could actually go in at the DNA level using something called CRISPR-Cas9, which is gene editing, and snip it out and make that cell no longer affected. Now, that's a big problem. You have to deliver the machinery, this CRISPR-Cas9, which is an enzyme, and it needs an RNA guide, in order to do that to the right cells in the brain. So challenging, to be sure. Now, that DNA gets made to RNA. This is the basic dogma of molecular biology. So the RNA also then has that expanded CAG repeat that's going to make glutamine if it gets turned into protein over here. So anything you could do to try to block that particular RNA from going any further would be a good thing. And that's where something like a oligonucleotide, you're going to hear about a strategy like that from Sarah, might be able to basically degrade this before it ever gets turned into protein. Or if it gets out here in the cytoplasm, it's something called RNAi, which also keeps that RNA from turning into protein. You're trying to avoid, or at least dial down, the production of the protein that has this yellow blob, which is that extended set of glutamines that were coded for by that CAG. That's the general strategy. And to be able to draw a diagram like this and say, we have things actually working in all those places is pretty amazing, because this would have been utterly hypothetical a few years ago. So with that, I hope I've given you a sense of the history of how we have, over the course of these 50 years that HDF has been in existence, been making steady progress. We know a heck of a lot about Huntington's disease, but I think we are still in the place where we need to push forward to take all that knowledge into what could be a treatment, or dare I even say a cure. And that means that while we have hope, we also need to have action. One of my favorite quotes from the poet Peter Levi, "Hope in every sphere of life is a privilege that attaches to action. No action, no hope." You are all here this evening to be part of that hope, but I hope also part of that action. And that way we can all feel that we are living up to what this expectation is that we need if we're going to have the privilege of hope to roll up our sleeves and get in there and figure out answers to this condition. I think we can do it. I think we're on a very interesting course to make that dream come true. And I'm delighted to have a chance to be here in New York to say those words to you, and then to listen to what my distinguished colleagues are going to tell you about what's really going on with Huntington's disease research right now today. Thank you very much.